Hello, my name is Mila, and today I will be talking about a project called Isolation, Quantification, and Analysis of Human Acid Phosphatase that I was working on with Eshra Tabassum over the past fall semester. So acid phosphatases are a group of enzymes that play an important role in various metabolic processes. Varying levels of APs can be indicative of various diseases in humans, such as leukemia and prostate cancer. Given their medical and biological importance, it is important to understand how we can study acid phosphatases, and this is basically the purpose of this whole project. Acid phosphatase was produced by introducing a piece of DNA of human acid phosphatase onto a bacteria. The bacteria produced a whole bunch of that enzyme. It was then crushed or lysed, and as you can imagine, this cell lysate, or this crushed bacteria, it contained a whole range of different proteins. So the first step we had to do was to separate our protein from this range of proteins. And we did this by immobilized metal affinity chromatography, or IMAC, and the sole principle of it is you introduce your cell lysate, you introduce all those different proteins onto a column with the stationary phase that contains nickel 2+, so positively charged nickel. And our protein is something that's called a his-tag protein, so it attaches to the nickel column and it stays there because it is attracted to nickel. So as we go through our several wash steps, all of the proteins that have no affinity or have very low affinity for nickel, they basically get washed away. And in the final elution step, we introduce something that's called imidazole, and it competitively replaces, so it comes in place of our acid phosphatase, and our acid phosphatase washes through and we can actually collect it in our elution fractions. So during IMAC, we collected 9 fractions, and then we measured them all with a spectrophotometer. So from this experiment here, we can see this is our elution fractions, and the fraction with the highest absorbance is the fraction with the purest acid phosphatase. And we determined that that for us was fraction 8. We then performed something that's called SDS-PAGE, or just gel electrophoresis, and the principle of it is we loaded all of our different fractions, and all the different proteins in those fractions, they separated based on their molecular weight. And we also have this molecular weight marker that we loaded to determine all the different molecular weights produced. So this basically confirmed that fraction 8 does indeed have our acid phosphatase, and it's the purest fraction because it only has one band. And we also determined that the molecular weight of our acid phosphatase is 17 kilodaltons, which is pretty close to 20 kilodaltons that we were expecting, and we got this value from bioinformatics. We also determined the concentration of acid phosphatase in fraction 8, and it was determined to be 420 micrograms per milliliter. We then went ahead and performed something that's called a Bradford assay. So that's a colorimetric assay based on color. We could determine the concentration of our acid phosphatase, the actual concentration, as opposed to the predicted one. So in this calibration plot, from this calibration plot of BSA, which is a similar protein to AP, we could determine that the concentration of our enzyme of interest was actually 191 micrograms per milliliter, which is significantly lower than what we were expecting. And our main source of error, we believe, is the fact that AP decomposes over time. Our last experiment was the enzymatic assay to determine whether or not our enzyme actually works. And the reaction in which we were testing it is basically hydrolysis of this species into paranitrophenol, and then this paranitrophenol, it actually produces this yellow species, the phenolate species, in the basic media. So we performed this reaction on several enzyme concentrations, and we actually got some yellow color, which means that our enzyme works. And we determined that the reaction velocity is actually linearly proportional to the concentration of acid phosphatase, which is something that we were expecting. Overall, in conclusion, we have been able to successfully isolate our acid phosphatase, quantify it via several methods, and study its enzymatic activity. So overall, the experiment was a success. Thank you very much for your attention. It was a pleasure working on this project.